And the question is, study the following statements regarding lactic acid bacteria LAB which are used to convert milk into curd. The options are as follows. They produce acids that coagulate and partially digest the milk proteins. A small amount of curd added to the fresh milk has an inoculum contains millions of LAB which at suitable temperature multiplies and converts milk into curd. Conversion of milk into curd improves its nutritional quality by increasing vitamin B12. LAB may result in acidity in the stomach of human being. Which of the following statements are correct? 1, 2, 2, 3, 1, 2, 3 or 1, 2, 3 and 4. The answer for this is C, which is 1. They produce acids that coagulate and partially digest the milk proteins. A small amount of curd added to the fresh milk as an inoculum contains millions of LAB, which at suitable temperature multiplies and converts milk into curd. Conversion of milk into curd improves its nutritional quality by increasing vitamin B12. Let's know how. Now, microbes such as lactobacillus and others, commonly called as lactic acid bacteria, which is LAB, grows in milk and converts it into curd. Now, during the growth, such bacteria produces acids, mainly lactic acid, that coagulate and partially digest the milk protein. A small amount of curd known as starter. Small amount of curd known as starter is added to the milk and kept at suitable temperature, which lactic acid bacteria multiplies in millions and milk converts into curd. That also improves its nutritional quality by increasing vitamin B12. It also checks growth of disease causing microbes in the stomach. And the next question, which of the following antibiotics is not correctly matched with the sources from which it is obtained? Antibiotic sources penicillin, bactracin, chrysophyllin, streptomycin. Sources Penicillium, Chrysogenum, Bacillus licheniformis, Penicillium rhizophilum, or Bacillus gracius. Now, the answer for this is D. Streptomycin, Bacillus gracius. Let's know how. Now, streptomycin is obtained from Streptomyces gracius and not Bacillus. And not Bacillus gracius. Now, it is found useful in meningitis. The next one is pneumonia, tuberculosis, and local infections. So, streptomycin is obtained from Streptomyces gracius and not Bacillus gracius. It is found useful in meningitis, pneumonia, tuberculosis, and local infections. Let's move on to the next question. Which of the following organisms is used in the production of beverages? The options are as follows. Penicillium notatum, Saccharomyces cerevisiae, Aspergillus niger, or Clostridium botulinum. The answer for this is Saccharomyces cerevisiae. Now let's know how. The yeast Saccharomyces cerevisiae is also known as very important brewer's yeast. It is known as BREWERS yeast, which is used for fermenting malted cereals and fruit juices to produce ethanol, which is ethyl alcohol. Microbes, yeasts in particular bacteria have been used for the production of beverages such as wine, beer, whiskey, brandy or rum. Is used in production of beverages. Beverages are wine, beer, whiskey, brandy and rum. Now these are the microbes used for the production of the beverages, which was the organism Saccharomyces cerevisiae. And the next question. In the sewage treatment, bacterial flocks are used and allowed to sediment in settling tank. This sediment is called as inactivated sludge, activated sludge, primary sludge or secondary sludge. The answer for this is B, which is activated sludge. Let's know how. Activated sludge is formed during secondary sewage. It is used during secondary sewage treatment. It possesses flocks of decomposer microbes and formation of activated sludge requires aeration. So, activated sludge is formed during secondary sewage treatment. Now, it possesses flocks of decomposer microbes. Formation of activated sludge requires aeration. Let's move on to the next question. During the primary treatment of sewage, solid particles that settle down are called flocks, primary sludge, activated sludge or anaerobic sludge. The answer for this is let's know how. Now, primary or physical treatment is the process of removal of small and large floating and suspended solids from sewage through two processes of filtration and sedimentation. First, floating and suspended matter is removed. Through sequential factorization filtration with progressively smaller pore filters. Now, this sediment is called as primary sludge, while the supernatant is called effluent. Supernatant is called as effluent. The primary sludge traps a lot of microbes and debris It is subjected to composting, landfill, and anaerobic digestion to produce biogas and manure. So, during the primary treatment of sewage, solid particles that settle down are called as primary sludge. Now, the supernatant is effluent. Now, first floating and suspended matter is removed through sequential filtration and with progressively smaller pore filters. Now, this sediment is called as primary sludge. So, it is subjected to compositing, landfill, and anaerobic digestion to produce biogas and manure. Let's move on to the next question. Which of the following bacteria is present in the remew of cattle? The options are as follows: Azotobacter, Rhizobium, Methanobacterium, or Azospirillum. Now, the answer for this is C, which is Methanobacterium. Let's know how. Methanobacterium is found in the rumen. Now let's know what is rumen. Given here, let's know what is rumen. Now rumen is a part of the stomach. In cattle. Rumen is a part of the stomach in cattle. So, Methanobacterium is found in the rumen, which is a part of the stomach in cattle. Now, a lot of cellulosic material is also available in the rumen. In rumen, these bacteria helps in the breakdown of cellulose and plays an important role in the nutrition of cattle. Now, how the digestive system in human beings are important? Same way, rumen, a part of the stomach in cattle, is extremely important for the digestion of food in cattle. And they have asked you that which of the following bacteria is present in the rumen of cattle, and the answer for this is Methanobacterium. Methanobacterium is found in the rumen, a part of the stomach of cattle. A lot of cellulosic material is also available in the rumen. In rumen, these bacteria helps in the breakdown of cellulose and plays an important role in the nutrition of cattle. Let's move on to the next question. Biogas is produced by aerobic breakdown of biomass, anaerobic breakdown of biomass, with the help of methanogenic bacteria, or both B and C. And the answer for this is B and C, which is biogas is produced by aerobic breakdown of biomass, anaerobic breakdown of biomass, or with the help of methanogenic bacteria, or B and C. Now the answer for this is anaerobic breakdown of biomass and helps the methanogenic bacteria. Let's know how. Now the biogas is a methane rich fuel gas produced by anaerobic breakdown of digestion of mass with the help of methanogenic bacteria. Biogas is made up of methane, which is 50 to 70%, carbon dioxide, which is 30% to 40%, with the traces of nitrogen, H2S and H2O. Now what is CH4? Methane. Now biogas is a methane-rich fuel gas produced by anaerobic breakdown of digestion of biomass with the help of methanogenic bacteria. But let's know what is biogas first and what does it consist of. Now biogas is made up of methane, which is a 50 to 70%, carbon dioxide, which is 30 to 40%, with the traces of nitrogen, hydrogen sulfide and hydrogen. And the question is, read the following statements and select the correct option. Statement 1. Biocontrol refers to the use of biological methods for controlling plants, diseases and pests. Statement 2. Use of biocontrol measures which greatly reduces our dependence on to rich chemicals and pesticides. Now this question is just type of assertion and reason. They give you two statements. They'll give you two statements and the options will be as follows as this way. Both statements 1 and 2 are correct and statement 2 is the correct option of statement A. Both statements 1 and 2 are correct, but the statement 2 is not the correct explanation of statement 1, or statement 1 is correct and statement 2 is incorrect. Both statements 1 and 2 are incorrect. The answer for this is B. Both statements 1 and 2 are correct, but statement 2 is not the explanation for statement A. Both statements 1 and 2 are correct, but statement 2 is not the correct explanation of statement A. Biofertilizers are some bacteria and cyanobacteria. Fertilizers formed by blowing in barcine, fertilizers obtained by decay and dead organisms, fertilizers prepared by mixing cattle dung with crop residues. The answer for this is some bacteria and cyanobacteria. Let's know how. Biofertilizers are organisms which bring about nutrient enrichment of soil and it enhances the availability of nutrients to crops. Microorganisms which acts as biofertilizers are bacteria, cyanobacteria, blue-green algae, which is also called as cyanobacteria, the mycorrhizal fungi. The mycorrhizal fungi, bacteria and cyanobacteria have the property of nitrogen fixation, while mycorrhizal fungi, the property of nitrogen fixation will preferentially withdraws the minerals from organic material for the plant with which they are associated. Now they maximize ecological benefits and it minimizes the environmental hazards. Environmental hazards means pollution, which causes pollution, whether it's soil pollution, air pollution, nothing like that. It's just a biofertilizers, which is good for nature and it balances the cycle between ecological benefits. And the next question is, biofertilizers are the living organisms which living about soil nutrient enrichment, maximizes the ecological benefits, minimizes the environmental hazards, or all of these. Now the answer for this is all of these. As I said in the last question, it maximizes the ecological benefits and minimizes the environmental hazards. And let's know how. Biofertilizers are microorganisms which brings the nutrient enrichment of soil by enhancing the availability of nutrients to crops. Microorganisms which acts as biofertilizers are bacteria, cyanobacteria, which is also called as blue-green algae, and also mycorrhizal fungi. Now bacteria and cyanobacteria have the property of nitrogen fixation, while mycorrhizal fungi preferentially withdraws the minerals from organic matter for the plant with which they are associated. They maximizes ecological benefits and minimizes environmental hazards. And the next question, in a microbiology laboratory, the technician uses heat to sterilize the nutrient solution, which is used to grow a fungus. Now, the heating system broke down. He sterilized the solution by passing it in a sterile environment through a sterile filter with a pore size of 0.2 micrometers. When the fungus was grown on the filtered nutrient solution, it stopped throwing and growing and looked unhealthy within a few days, with statements with the most likely explanation for the observed effects on the fungus. The nutrient solution contained a virus. Heating makes the glucose in the nutrient solution more digestible. Filtering removed one of the larger nutrient molecules. The nutrient solution contained a bacterium that was pathogenic to the fungus. The answer for this is the nutrient solution contained a virus. Now let's know how. This is a very, very important question. These type of questions are mostly asked. So the nutrient solution could have contained a virus. How? It contained a virus which passed easily through the sterile filter of pore size of 0.2 micrometers. Yes. And this has caused retarded and unhealthy growth of the fungus. Hence, the nutrient solution contained a virus. Microbe used for biocontrol of pest, butterfly, caterpillariasis, Saccharomyces cerevisiae, Bacillus thuringiensis, Streptococcus streptococcus, Thiroderma spirulina. The answer for this is Bacillus thuringiensis. Let's know how. Now, biopesticides are those biological agents that are used for control of weeds, insects, and pathogens. The organisms used as biopesticides are viruses, bacteria, pathogens, protozoa, and etc. Fungi and mites also. Now, some of the biopesticides are protozoa, fungi, and mites. Some of the biopesticides are being used at the commercial scale. Most important example is the soil bacterium. Bacillus thuringiensis PT. Spores of this bacterium produces the insecticidal cryprotein. Therefore, spores of this bacterium kills the larvae of insects which eats them. In batch fermentation, substances are added to the system at all once and runs until product is harvested. Nutrients are continuously fed into the reaction and the product is symphoned off during the run. New batches of microorganisms are screened for increased yield. Small scale production is used to synthesize product. And the answer for this is A. That is, the substance added to the system at all in once runs until the product is harvested. The vitamin whose content increases following the conversion of milk into curd by lactic acid bacteria is vitamin C, D, B12 or E. The answer for this is C. Vitamin B12. Let's know how. Now the curd is more nutritious than milk. As we know, milk contains lactic acid. It is good for your muscles. It contains proteins, but curd contains minerals, vitamins, and more nutritional values rather than milk. It contains a number of organic acids and vitamins including B12. Methanogenic bacteria are not found in rumen. Now what is rumen? As I said before, rumen is a part of the stomach in cattle, which is very important for the digesting of food in cattle. Rumen of cattle, gobar gas plant, bottom of waterlogged paddy fields or activated sludge. The answer for this is A. That is rumen of cattle. Let's know how. The sediment of settling tank is called as activated sludge. A part of it is used for inoculum in aeration tanks. The remaining is passed into a large tank called as anaerobic digesters. Now, methanogens are strict anaerobes. Nutritionally, they are autotrophs, which obtains both energy and carbon from decomposition products. They occur in marshy areas where they convert formic acid and carbon dioxide into methane with the help of hydrogen. This capability is commercially exploited in production of methane and gas fuel inside the gobar gas plants. For example, Methanococcus methanobacterium. So, some of the methanogenic archaebacteria live as symbiosis in the stomach of herbivorous animals that chew their curd. Ruminants, for example, cow, buffalo, these archaebacteria are helpful to the ruminants in fermentation of cellulose. The warm, water-logged soil of paddies provides ideal condition for methanogenesis, and though some of the methane produced is usually oxidized by methanotrophs in the shallow overlying area, vast majority is released into atmosphere. Methanogenic bacteria is not found in activated sludge. Instead, activated sludge contains wastewater treatment generates a large quantity of sludge which can be treated by anaerobic digesters, fog, caramel or chemicals or oxidation pond. The answer for this is anaerobic digesters. Let's know how. The statement of settling tank is activated sludge. The part of it is used in inoculum. Wastewater treatment generates a large quantity of sludge which can be treated by anaerobic digesters, floc, chemicals, or oxidation pond. The answer for this is anaerobic digesters. Okay, the question is, the primary treatment of waste water involves the removal of dissolved impurities, stable particles, toxic substances, or harmful bacteria. The answer for this is B, which is stable particles. Let's know how. Now, the primary or physical treatment is a process of removal of small and large floating and suspended solids from sewage through two processes of filtration and sedimentation. First, floating and suspended matter is removed through sequential, then kept in large open settling tanks, where grit, which consists of sand, slits, and small pebbles, settles down. Aluminium or lime sulfate is added in certain places for flocculation and settling down the solids. The sediment is called as primary sludge, while the suspendent is called as effluent. Now, effluent is a suspendent. Let's move on to the next question. Which one of the following alcoholic drinks is produced without distillation? Wine, whiskey, rum, brandy. The answer for this is wine. Let's know how. Now, wine and beer are produced during distillation. Without distillation, it is without distillation. Whereas whiskey, rum, and brandy, these three beverages are produced by distillation. And the next question, the free living fungus Trichoderma can be used for killing insects, biological control of plant diseases, controlling butterfly caterpillars, or producing antibiotics. The answer for this is biological control of plant diseases. Now, a biological control being developed for use in the treatment of plant disease is in the fungus Trichoderma. Trichoderma species are free living fungi that are very common in the root ecosystems. They are effective biocontrol agents of several plant pathogens. The answer for this is biological control of plant diseases. Which one of the following is not a nitrogen fixing organism? Anabaena, nostoc, azotobacter, or Pseudomonas? The answer for this is Pseudomonas. Let's know how. Pseudomonas is not a nitrogen fixing organism. Pseudomonas is a saprophytic bacterium. Pseudomonas are used for biodegradation of organic pollutant like petroleum spillage. Petroleum, it's very important. Spillage. Now, what is Azotobacter? Azotobacter is a free living organisms. Anabaena and nostoc are free living and nitrogen fixing cyanobacteria. So the question is, which is not a nitrogen fixing? This is the answer. The residue left after methane production from cattle dung is burnt, buried in landfills, used for manure, used in civil construction. The answer for this is C, used as manure. Let's know how. Biogas is a methane-rich fuel gas produced by anaerobic breakdown of digestion of biogas with the help of methanogenic bacterium. Biogas is made of methane 50 to 70%, 50 to 70% methane, 30 to 40% with traces of nitrogen, H2S, which is nitrogen sulfide, and nitrogen. The effluent and residue left after the fermentative generation of biogas is rich in minerals, lignin, and a part of cellulose. It is an ideal manure. Big holes in Swiss cheese are made by a machine, a bacterium that produces methane gas, a bacterium producing a large amount of carbon dioxide, or a fungus that releases a lot of gases during its metabolic activities. The answer for this is C, a bacterium producing a large amount of CO2. Let's know how. Ripened cheese is prepared from unripened cheese by first dipping in brine, wiping, and then maturation with different strain of bacteria and fungi. It takes at least 1 to 16 months for ripening. Large whole Swiss cheese is ripened with the help of carbon dioxide causing holes. CO2 causes holes in Swiss cheese, keep this in mind, and the bacteria which causes carbon dioxide is Bacterium Propionibacterium shermani. This is very important example. Big holes in Swiss cheese are made by a bacterium which is called as Propionibacterium shermani, which produces a large amount of carbon dioxide which causes holes. Methanogens do not produce oxygen, methane, hydrogen sulfide, or carbon dioxide. The answer for this is it does not produce oxygen. Let's know how. Biogas is a methane-rich fuel gas produced by anaerobic breakdown or digestion of biomass with the help of methanogenic bacterium. Biogas is made up of methane, which consists of 50 to 70%, carbon dioxide, which consists of 30 to 40%, and the traces of nitrogen, H2S, and H2O. It is an ideal manure. The informative generation of biogas is rich in minerals, lignin, and a part of cellulose. Methanogens do not produce oxygen. Match column A with column B. Ladybird, mycorrhiza, biogas, biological control, methanobacterium, trichoderma, aphids, or glomus. And the options are as follows. Ladybird with aphids, mycorrhiza with glomus, biogas with methanobacterium, and biological control with trichoderma. 1. Ladybird is aphids, which is C. Mycorrhiza is glomules, which is D. Biogas, which is methanobacterium, A. 4th, biological control is trichoderma.